And we're back. My name is Jeremy Horowitz. I'm a PhD student at James Cook University in Townsville, Australia. I'm affiliated with the Centre of Excellence for Coral Reef Studies and the Museum of Tropical Queensland. This is the Schmidt Ocean Institute, ROV Falkor, ROV SuBastian, Expedition FK200802. And this is dive S0372. This is the second dive on North Flinders Reef out in the Coral Sea. Joined in the control room, Chief Scientist Brendan Brooke, Marta McNeil, and Andrew Carroll, who are annotating what we see, and they'll also chime in from time to time throughout the dive. And the ROV pilots are Chris, J-Rod, and John. And the purpose of this expedition is to map seamounts, canyons, and reefs out in the Coral Sea, and collect the rocks and biology to understand how the habitats came to be and what lives within them. This is the second dive on this reef, and we'll try to collect some more biology and rocks. And we're gonna start where we ended bathymetrically on our last dive at 343 meters. So what we're looking at right now is seafloor, and I'll just zoom in on this coral that we opportunistically landed in front of. So it's a whip coral, but that doesn't tell us what order it is. We need to look at the tentacles, and I can already see that it's an octocoral. So this guy has 8 tentacles. And this is one of the target orders for this cruise, so we will certainly collect it. So we'll just get a good close-up shot of the polyps. And can we get lasers?
Fish. Definitely a fish.
There's a fish nearby. Lasers are on. I'm just going to zoom out so the lasers are in view. Okay, there they are. So that's 10 centimeters apart between the 2 green laser points. So, I don't know, extrapolate from that. All right, so we're gonna take a push core sample first, and then we'll take a fragment of this octocoral, and then we'll look for some more good stuff to collect.
You ready? How close are we? Are we going to hit it with this?
I think I'm on a ridge, so I'm going to push off anything, but I'll pick up. Well, you'll be able to get, well, let me turn that down a bit. It's right there. Come on, it's a little bit. You should be able to get 1 and 2, no doubt.
Okay. I'm ready for it.
Push it out. Yeah, especially so late. So you're fine. Really win. Alright, I will take over for a minute while you're doing this, and then yeah. Let's change that bed.
Thank you all, where it's bedtime. Yep, try to stay awake. It'll be a short dive, probably around 2 hours.
We want to go anywhere in particular?
Anywhere.
Coming down with the RV a little bit down.
Yeah, I'm just trying to.
So we have experts who couldn't make it on the trip. so they're home, but watching around the world. So when we come across corals and fish, we will ask them for their expert opinions on identifications.
I'll try another one.
Yeah. Maybe a scoop.
I can also do a scoop with this as well. Done it in the past.
The issue is whenever I took it out yesterday, it was kind of, it did not make it up too well.
The whole scoop wouldn't be
Well, I mean, it's going to be the same as the section.
Well, that's the movie, you can't put it in the,
Can't what?
we put it in the, it was the kind of, it's just coming out, then we got it on the deck, but actually,
Out in the open, right? No, that valve definitely open. I'll let you back out. That's pushing.
I'm pushing
Okay.
down. I'll
Core
find out.
attempt number 2.
No.
Right. So, definitely not going to hold in there.
I've got the bag scoop.
Right.
Probably work.
I can try one scoop with this
Yeah.
in the bag as well. What do you think?
What's the same? That's the same one that didn't work yesterday. Number 1 didn't work. Which one
They
did?
they fixed this one.
Yeah.
Yeah.
I mean, I could try a different one as well.
So is it that it's fine?
How deep of
It's,
sediment do you
Just
want?
a few centimeters.
We're pushing the, let me bring the torch in it, make it easy for you to see, or you got it? Okay. We saw the valve open, so it was producing a vacuum. It's just that it's such a granular, fine sediment that
I think we've done this before.
just
I have
That's great.
Yeah, okay.
we can put it.
work on.
All right.
I've done it once.
Close. All right.
One way or another, we got it in.
All right, you still want to do a scoop bag though, right?
I don't think we need to do that.
Yeah, that's probably enough. I think that's
You think that's going to be enough?
yeah. Thank you.
Thank you.
Oops, not on the phone.
Nope. We'll have it yesterday. We went in to get it on there.
You weren't able to get it back in the pump?
It wasn't on there when I took it off of the rack.
Hello Queensland, hello Perth, Netherlands.
All right. What else do you want to collect?
We'll collect this coral. We don't need the full colony, so 5 inches should do.
Don't know if you reach it.
Can we just zoom in for a minute first? I'll just focus on the polyps.
Yeah, I got pulled away whenever I pushed out, so I'm gonna push back in and get you closer.
All right.
Of course, now I have to do it. Now I just push it in. Alright, that's what it is. Alright, it's like this.
All right. So we're just gonna zoom in here and look at what we're about to collect. So as I said before, this is a soft coral, and we'll try to zoom in on the polyps. Maybe the auto zoom will do it. So what we're going to do with this sample, as with all of our other samples, we're going to describe the suite of morphological features of each of these specimens, and we're going to also extract the DNA from these specimens, and we'll sequence that DNA. And based on these two lines of evidence, we're going to understand, one, what species it has traditionally been called based on morphological features, and two, its evolutionary history, how it relates to closely related species. And with these two lines of evidence, we're going to better understand how this species came to be and its range extent, where else it's been found, and its taxonomy. So every specimen is going to be a little bit different than the holotype specimen, or the first specimen that was collected from which the description was made. So we'll take this fragment, describe its morphological features, and sequence the DNA, and then specialists within the soft corals will take it from there. So I would just focus in, if it's possible. I'll just try to get one.
So we got it. All right.
Yeah.
On the left, it's clearer than on the right.
Yeah, that's okay. So it looks like there's maybe 3 rows of polyps. I'm not a soft coral expert, but I assume it's a morphological feature that's used to identify the species. All right, let's grab a piece.
Okay, so about that much right there?
Yeah, that'll do. And as with many corals, so this coral is colonial, which means each one of these polyps is an individual animal. So for this specific colony, we didn't kill it, we just took the top off of it, so it'll grow back.
Yeah, we'll give a good lift over here. Yeah, got it.
Got it.
Hello again. So for those who are just joining in, we are exploring the Coral Sea. We are looking at corals and sponges and the geology on specific reefs. And so we're doing ROV work, which is what you're seeing now, and also AUV work. So we're mapping the sites, describing what species live within these habitats. And afterwards, the geologists and biologists are going to go home and work on these specimens that we collect to better understand these long-standing questions.
Is this a floaty or a sinker?
I'd call it a neutrally buoyant fragment.
Box one. Yeah, we'll ask that question if you allow when we have a coral photo. We'll ask that question if you allow.
Yeah. Well, yeah, I don't think anything's going to really float that we'll collect. Yeah, experience says otherwise.
It's not so much that they float, they're just so wind
John,
that any current
you want a
is
second?
taken away.
Right. Yeah, so as I said earlier, this is going to be a short dive. We got in the water a little bit later than expected. This is only our second dive, so we're still getting into the groove. But luckily, we've got 3 weeks ahead of us of ROV dives almost every day. So we're going to be exploring for the first time this part of the Coral Sea. and you guys are able to watch along with us. Are you grabbing that rock?
Do you want tape rock?
Yeah. Sorry. Marty. Want this rock?
No, don't want to lose rock things.
No.
Do you not want it?
No. We want
Let's get out of
we want a bit from in
Photo
situ,
of-
don't we, Brendan?
What's
Yeah,
that?
from an outcrop, but then
Yeah.
Look at that.
oh, okay, so it has
So we're not going to take this rock,
worm tube.
but you can see lots of little things living on it.
That should be ABs. Do you see when the local one?
You can see tubes, worm tubes,
That would have been one.
little coral. I'm not sure exactly what it is.
Hang on.
We did change.
All right, so the plan now is to head forward, and there's a big rock outcrop,
Okay.
and we'll pull a piece off of that.
Sounds like a great plan.
You like that rock, didn't you?
No, I wanted to make a little, what do they call them? Or whatever. A little rock statue. I was here.
Alright, so you want to go forward, you said?
Yeah, please. Okay, so we're gonna grab a rock sample and then do a bit of exploring, going shallow. We'll do one transect, a 500-meter transect, and that'll take us near the surface. That's the plan for today.
Eating down? Yes.
All right, so I think
Brendan, you can choose this rock where you want it.
You missed that.
Wow.
You saw that rock?
I think that might be what the plan is, yeah.
Yeah.
so. The reason why that first rock wasn't ideal was because we didn't know where that rock came from. So what we're looking at now, we'll sample a bit off of this, and that'll give us more insight into this habitat. And I see a fish, a few fish in these hidey holes. I don't know if Kai's online.
That's probably up.
There is no option two.
Coral up there.
Okay, I'll put that in right now.
You said a little coral or
There's another coral at the bottom right as well.
rhizome? Fake corals? What are fake corals called?
Do you want a piece of this rock?
Yeah, we could do that.
Do you want to look around it first, or just take a piece of the...
Yeah, let's have a little bit more of a look, but it seems to be a significant deposit.
Would you like anything attached to said rock?
Pardon?
Would you like anything attached to the rock?
Depends what it is. That's coral, yeah.
Like that coral?
Yeah.
That's pretty. Hello.
Buddy. Black ball.
Oh, look at this. We got Anemone and Gargonian down in the bottom right. And lots of little, I'm going to say bryozoans on the rocks. But first we'll take the rock sample
Okay.
and then we'll look at these different corals and grab a few.
We're gonna come back up to the rockier, rocky part.
Yeah.
So what's cool about these underhangs is a lot of these hard corals will grow underneath them, which makes it hard for people to discover them. So a lot of them are unknown. And even in the shallows, a lot of them are azooxanthellate, which means they don't rely on sunlight to grow. So in the shallow end and the deep, very little is known about these corals. So we have the opportunity to collect underneath these overhangs. So we'll give that a go after we've finished collecting this rock sample.
Yeah, the one touch is what I had it on. I did this in and out. It was not a good idea.
Yeah, it's like part of the White House.
Let's piece of this one of these rocks here.
One of these rocks?
What's that?
Do you want one that's loose or do you want one broken off?
Broken off, please.
All right, I'll get you a little closer.
See a whip coral,
I've really got to concentrate on what I'm doing.
a lot to look at, a little overwhelming.
We can get something out of there.
Try.
All right, I'll switch
I'm
that.
gonna pour it on this one.
Corner.
Yeah, just because it's a better angle. That's one. And I'm right there. Coral cutters too.
Damage.
Hello, California.
All right, I'm sitting here gingerly, so if you have to fight it, like I'm gonna have to.
How big? You looking like tennis ball size?
Yeah.
Yeah.
Yeah, similar to yesterday's, I think.
No, not that big roll.
You didn't like the one from yesterday?
The one, it was too big. It was beautiful, but it was big.
And so actually, we took a rock sample yesterday, and after bringing it in the lab, we found a lot of corals on it, although we couldn't see it from the ROV footage. So it's also exciting to look at the rocks before they're scrubbed. We find little cup corals in the rocks and bryozoans and worms.
Try to break off this piece, maybe.
That's all right. I'm very gingerly rusty, so there's a way I might have to like fly into it a bit. About right there. Below it.
That little dip right here?
Yeah, right there.
The actual full one?
That
Little nothing.
guy right there.
Cool like that.
That's a good looking rock.
Do you like that one?
Accessibility is what it's
Do you
amazing.
like this rock or do you want something different?
Not too excited.
I mean, I can break it off and then we can have a look at it.
Yeah.
Maybe it'll change your mind. Wanna go?
I just need to actually sit down.
Here, have a look down right here.
ROV control breach.
Go ahead, Rich.
Hey, do you want me to move to that new waypoint?
We're good for right now, actually. We're gonna stay by where we are.
Oh, okay, stand away.
Those rocks are.
All right.
Who would have thought rocks are hard?
Hold on.
Don't worry, as soon as I grab on, you won't go anywhere. That's a really
Alright.
small piece of rock in there. It's not going to have anything useful to you. That's just a shark. Right? You agree with that, or do you want that?
You reckon, Marty?
It'll be better than nothing if we
Yeah,
don't
I
get
mean,
anything else.
I can get you something else. I think we just have to might move up a little bit.
Yeah.
Yeah.
I can try this little ear piece. Okay. I really want to. Are you holding down pretty tight there?
I can.
All right. Ready?
Yeah.
There. Got the ear piece just fell off.
What? You got something now?
I think that'll.
Oh, no, it's this stuff right
There
here.
it is.
How's that? Is that a little bit better?
All right, want to put it in
And
one?
it's a twofer.
All right, we got ourselves a rock.
Too much because it is two rocks. How's that, guys?
Happy with that, Marty?
That's fine, thank you. Keep it.
All right. Bob, box one again.
Looks like a little brittle stars coming along for the ride. Maybe a sponge or well, we'll have a look when we get it back on deck.
Thanks, mate. Yeah, watch those. Thanks, buddy.
Okay, once this rock
That one A.
is in the bio box,
It's labeled as
we'll resume exploring.
E on the thing, but however, it's I don't know what.
Yep, thanks.
Yeah, let's call it 1A.
Yep.
Upward is touch. Back half. That's good stuff like that.
Thank you.
All right,
Nice job.
so
Off the backboard.
Alright, more rocks.
I think we're
Get going, yeah.
Yep.
Okay, so we'll maneuver over to the start slope point and then
okay.
I'm going to let stuff with it. Where are you driving?
ROV bridge.
Yes, you can start moving to that waypoint now.
Yep, no,
All right,
Brown
ready to
Reef,
leave?
I can say something.
Yeah. Whatever you want.
Okay, when, does that want me to look at you or?
Okay, so we're gonna move to the start, the planned start for this survey. Be there shortly.
Very steep margin, water depth of 330 metres. Here, the sea is dominated.
See a lot of life.
Consolidated carbonate sand. There's also
Potentially some sponges in the background. Sea anemone.
So, what appears to be limestone, so we've just collected a sample of that, and also a sample
Going to
of
dive.
sand.
Are you going back to the start or are you going toward?
Let's go to the start. I thought we were doing the transect thing. 700 meters.
Not yet.
Oh, we're not doing that yet. That was my bad. Okay.
That's right. Actually, where we are now is interesting. If we look at the top of this heading,
Moving sediment that's produced
Top of this.
up on the reef,
yeah.
down the margins, and handling the
Yeah, I thought we were going back.
old and decimated itself. Former reef.
So up there, there's a bunch of stuff. I think up there.
That's great. My misunderstanding.
That's right. We'll get there.
We brightened up a little bit.
Terry, this is all stuff that's tumbled down.
Yeah.
All right. So we're gonna head towards this group of corals and what looks like a sponge. Really cool looking sponge. There's a whip coral. So we'll try to get as close as possible and then decide what we're gonna collect.
That's real habitat.
Which I'm interested. If we can get to this.
My gal's been buddy back on.
All right, we got a lot to look at.
Can we sit down so you can make it on?
We're definitely going to want to collect this here, and this, and that, and that. It's a good location.
I like that.
Yeah. So, oh, and also this right here. So, yeah.
Okay, gotcha.
So there's a lot here. So we'll take it one by one.
Okay.
Quite a bit of diversity in this little area.
Yeah, it must be that it's a bit on that rock top, so it must be getting some nutrients. Anemone. I'm not sure. Yeah, we'll have to look closely. So, what we're looking at right in front of us, that brown-looking coral, it looks like a Parentopathes. It's a black coral in the Schizopathidae. I'll need to look closer. Once we settle down, we'll take a zoom in. Yeah. So what we see here is 1, 2, 3, 4, 5 suborders of life in this small area. I guess what allows there to be such high diversity in this site, in this specific location, is that it's up on a ledge, so it might be getting lots of nutrients coming over the side, cutting across. You could see the corals are angled in the same way, I guess because of the current. So what we're going to do now is look at this black coral here. And again, you can see the two dots 10 centimeters apart to see how big it is. So it's pretty big. We definitely want to try to collect the whole thing. I'll just zoom in just to confirm. We'll just try to zoom in here.
Look down. Go look down it. Down towards the base. Then you'll get into it.
Yeah, look at that. That's awesome. So, yeah, looks like Parentopathes. So what distinguishes the species within this genus are the number of what we call pinnules, or repeated branches along the stem. So it'll be hard to identify the species until we look at it. And you can also see there's a lot of associated fauna on it. There's a Brittle Star here, maybe a few of them. Probably some crabs. Really beautiful. Okay, got a good view of it. So we're gonna collect it. This is the first Parentopathes we're collecting. We expect to see a lot of them, and really, there's probably many more species than are currently described. So this will be our chance to either expand the range of the species to this part of the Coral Sea, or describe a new species. And the reason why we're going to collect the full colony rather than just the tip is because what we use to identify the species, besides the pinnules, are what we call skeletal spines. So they're spines along the corallum or the colony, and they're very distinct. And they seem to be the most informative feature in separating closely related species. And those spines can vary from the top to the bottom. So in order to be sure of our ID, we're going to collect the whole colony. Additionally, there are what we call striatum at the bottom, at the base of the colony, and those are ridges. And some species have them, some species don't. We don't yet know if it is informative at all in telling us anything, but until we know for sure, we'll look at it. Yeah, there's bryozoans there. That's a bryozoan. A little hermit crab. A lot of stuff. Little anemones. This is a really amazing site right here.
Great job, guys.
Okay, we'll do one more zoom in. One last look.
Yeah.
So that's a nice shot. It's a good shot.
Oh, it's beautiful. So some of the things we'll look at besides the spines and the pinnules are also the polyps. So you could see, as we zoom in close, each polyp has 6 tentacles. So if you see groupings of 6 tentacles, that's one polyp. And so we will measure the length of the tentacles, the distance between two pinnules, and of course the rows of pinnules, which they tend to grow in spirals. So you can almost see it. This is kind of the backside of the coral. There might be 5 or 6 pinnules in a spiral row, but again, we'll look at it when we get it back on deck.
All right. I'll
Yeah.
try to get you that view that you were just talking about.
Right. Yeah, that's awesome.
Thanks, John.
Yeah, you can see it from the top.
Well, let me come out. Alright.
Yeah, this is actually amazing to see in situ the colony. Usually when you get it on land and then in ethanol, the morphology changes a little bit. So it's great to get this view at depth. Can we look head down on the top of the stem?
On the stem.
Yeah, so like turn it downwards.
So this piece right here, right?
Yeah.
That we're looking at?
Yeah, and then just focus on
Just the very head.
the foreground. I see a fish's shadow and coming into view. We'll look at that in a second.
I'm gonna go out all the way back and see
Okay.
You should just be able to go to manual and just turn it.
Oh boy, look at that.
Wow.
So now you can see the pinnules, how they spiral. Even with this amazing view, it's hard to tell. What we'll do is we'll cut one fragment off and then count pinnule by pinnule and see how spaced out each pinnule is. And again, a pinnule is a repeated branch. Oh yeah, look at that. Oh boy, that's pretty cool.
Okay.
Anything inside?
I don't see anything in there. Maybe there's something orange in there, right there. See it?
Oh yeah, there's something in there.
Little shrimp, maybe.
Yeah, that's amazing. No, that's the Brittle Star. So the Brittle Stars often use corals like black corals to elevate themselves off the substrate to pick out particles floating in the water. All right, let's get this in the box and then grab the next one.
It does look like I am gonna write a note.
Uh-huh.
He's gonna write a note with an old feather.
Oh, yeah, it's like a feather pen. Yeah, it's pretty amazing.
I'm gonna porch out an early show. That's what that's gonna do to me.
You don't have to go all the way. Just come out enough to where and then came back.
Yeah, I just don't even know what I'm sitting in.
If there's a segment left over, it absolutely can regrow. So each of those polyps is an individual animal, and as long as there's one remaining, it has the opportunity, the chance to regrow back into a full colony.
Porching out
So I guess we'll put this one in bio box 2, or do you want to make sure this thing can lay out completely?
You could put it in the box that's one full, not the back one.
Yeah.
That'll be the one on the starboard side.
The one that has the scoop in it.
Right.
Do you mean the long one at the back?
Oh no, not that one. The
Box
not.
3, 4.
The one on the starboard side forward.
Okay, you know what I mean.
The one that's got a scoop in it.
Ah, it doesn't matter. As long as it
I can
fits.
take the scoop out.
No, it doesn't matter.
How's that heavy?
Okay.
I will get it right one time, I promise. Nice.
Yeah, we got some excellent pilots with us today. Making this look real easy. Oh, there's so much to see here. Once we get this in the box, there was another one, yeah, right in front of us, that branching coral, brown.
Come out a little bit with the camera. There.
Bounce away. And then
So we can grab that right where we are.
yeah.
Fortunately, got that right.
All right. So what we're going to look at next.
Move the camera out a little bit, so it's gonna change. There, you're fine. You can do that. Just move the arm out.
All right. Let's see what we got here. All right. Actually, this looks like could this be a bryozoan or a soft coral? I'm not sure. I think we won't collect this one just because I'm not exactly sure what it is. I think it's a bryozoan. But
Old salty one.
so you see the cup coral and then the hard coral right here. Can we get that where we are, or do we have to move?
Also, the cup coral on the back behind, and then the white one.
This and
Middle
this.
top.
Oh, yeah. Maybe look up with a.
So we want to make sure we're collecting only what we're allowed to collect, which is why we'll leave this one alone.
Cut coral.
Right between those rocks. It's just like, looks worse than the sit. It does. I might zoom in a little bit.
So we're going to try to grab.
I think I can get it.
Get plenty of
So we've got J-Rod trying to grab the cup coral here.
I might be able to get that one.
stand by.
You want to get it before you zoom in on it, or
It's probably better just to grab it.
you telling me to stand by first?
Yeah, I was gonna give it takes so you can see what that was.
No.
I can speed up a little bit, maybe.
So for you guys just tuning in, we are about halfway through our dive, our 2-hour-long dive. So we have about an hour to go, and we're in North Flinders Reef in the Coral Sea, and we're just collecting corals. We came about a really healthy site, and so we spent a little bit of time here, but it's early in our trip, so we haven't really collected all the targets that we're looking for. So we'll take the opportunity now while we're here to grab these different suborders of corals.
What's that?
Is that that bad one?
So it's rocks are less covered in
Is that the crown sea star?
Yeah, I think so. I think these are and there's a sea star.
Is that the crown or something?
No, it's not a crown of thorn, just a thorny sea star. Okay, so we've repositioned and we'll try to grab this cup coral. Is it warm there? Yeah, it's pretty warm.
Temperature in the water is 16.5 degrees.
Underwater at 330 meters is 16.5 degrees Celsius. Pretty warm. I think, yeah. The brown branching one, I think it's a hydroid. It's beyond the scope of my expertise, but we've taken a picture of it, and Robin Cummings at the Museum of Tropical Queensland works on bryozoans. So she'll let us know if it's a hydroid or a bryozoan. She'll give us a better indication.
So John, I'm assuming that you watched this pretty intensely whenever you were at home. Same at home as it is out here, or is it over here?
So it's a very fragile coral, it's small. So J-Rod's gonna be careful with his collection. I have no doubt he'll scoop it up without a hitch. Yeah, the cup coral probably is alive. I mean, can't say 100%. But although there isn't color, these cup corals that usually are in crevices hiding from sunlight, they tend to not have color, so it's probably alive.
I saw it up.
It's tough.
Yeah, it seems like that smaller one was growing off the other one. It is no longer growing on the larger one. So, Tom Bridge at the Museum of Tropical Queensland was interested in this cup coral, but he'll have to deal with the next one we come across. That's how it goes sometimes. We could grab the other hard coral,
Which one?
the white,
Oh, you want the base?
yeah, that big one. Oh no,
No,
Sorry.
Oh, that bryozoan?
Use my trusty pointer.
Yeah.
So you can grab a fragment of it.
Just a fragment?
Yeah, that's fine.
These are the real brittle ones, though, right?
Yeah, do your best. It's okay.
Can we go with the angle, or you mean move? Yeah, you're sit.
The green thing on top of the rock is an anemone. That's as much as I can say about that green thing on top of the rock.
got your original on that, so
I do. So I can take it out of your picture.
These hard corals are pretty delicate, but look at that, we got a nice fragment.
I'm gonna zoom out of the sit a little bit. Did anything look wrong? Alright.
Okay.
Put it.
It can go in the same box as the black coral.
that one.
The right one.
The last one that we just done?
Yep.
Okay.
I'm gonna take a look at that while I'm setting up and the camera in if you want. Get a close look at it while I'm setting up.
Yep. I'll just take a quick zoom in. We'll just try to focus.
Yeah, the auto to do it. Alright, give me a second.
Are you a man?
Yeah.
I want that on.
Here we go.
Yeah, look at that. Okay. Beautiful.
Is that good?
Yep.
Ready to start?
Heavy?
Yep.
Alright. And
Okay. In the box it goes.
coming up.
Okay. Yeah.
What else do you want to grab?
I'm just gonna take a quick.
Yeah, you got the camera.
All right, I'm just gonna look. We've been here for a little bit, so I'll just see if there's anything else of particular interest. Looking at this little guy here. Oh, sorry.
Yes, you got lock on right there. Sounds good.
All right. So this seems to be like another Parentopathes. So we won't need to collect that. Not here.
Yeah, I'll jump in. Or do you want to jump in with dibs, or do you want me to? You want to do it?
All right. So can we grab the top half of this soft coral here?
Top half this one.
Yeah, I'll just zoom in
Yeah,
for a view.
that's up to date. Cool.
So this looks similar to a soft coral we collected yesterday.
How about the change?
Top half of that.
We're gonna pick that one.
Carter, you're over there doing double duty.
So, all right, we have Adam and Ben
Logging and answering questions.
now on the ROV controls. Shift change. So, there's also focus. Okay. Yeah, there's a lot going on in this little microhabitat here.
Yeah.
Yeah.
Are you finished with videoing, Jeremy?
Yeah, you can. We'll just grab the top of this soft coral, and then we will move on. Jason, you see sponges? Oh. Oh, the little black things?
A bit more, Jeremy?
That's okay. That's good.
Good.
Yeah, it's enough. Okay, we'll look at that quickly before we move on. Yeah, there's never enough time.
These are old scissors.
Yeah, they're all dull.
Nice.
The batter, I think, is really strong.
There we go.
Bottom box 2's okay?
Sure. Yeah, that's fine. Okay, and you go with your friends.
Jeremy, sorry, was that black coral or soft coral?
Soft coral.
Wait, in the corner.
Billy, to answer your question, what benefits does researching coral? Is it helpful to understand health cares? There is research going on. I think particularly with sponges, with regards to medicine. But for our purposes, the reason why we're collecting biology is to understand what species live in this habitat. And that's important to understand because in order to protect something, you first have to know what it is, where it is, and how far it extends. So after this research, we'll have a pretty good idea about how many species live in this environment, where there are common species, where there are rare species. And then with our limited conservation resources, conservation dollars, and physical blocking off of areas from fishing and mining, this information gives us more. It gives us more information to make more informed conservation decisions. So, okay, we could stay here all day, but I think it's time to move on. So we'll head towards where we will start our transects. And we will try to do a continuous 500-meter transect, unless we find something spectacular. And yeah, Jason, I know what you're talking about with the potential sponges in the background. We've got 3 weeks, so we'll definitely get to it.
That's the waypoint 2 you want to head to? Is Andy going to say something about the transit?
Yeah. Once we start it.
Okay.
I'm going to pass over the mic to Chief Scientist Brendan Brooke.
Yes, good afternoon everyone from the Schmidt Ocean Institute's research vessel Falkor. My name's Dr. Brendan Brooke. I'm the Chief Scientist on this Seamounts, Canyons and Reefs of the Coral Sea research cruise. If you'd like to know more about our survey and the scientific questions we're addressing, then please have a look at the Schmidt Ocean Institute's website. We have this research cruise has a web page on the website, and we've got quite a bit of information in there about the background science as well as the team members, and some blogs about how we've gone about planning the survey, and our own individual roles in the survey as well. So that might be of interest to some of you. So you're watching dive 372 of ROV SuBastian, and currently we're at a depth of 322 metres on the northern margin of Flinders Reef in the Coral Sea Marine Park. So that this location is approximately 250 kilometres east of Cairns. And on this dive, we're collecting both high-resolution video footage that you're watching at the moment, of this really steep margin of the northern end of Flinders Reef. So in a minute, we're about to commence a 500-metre transect where we maintain a consistent distance above the seabed and a consistent attitude and speed as we move over the seabed to enable us to quantitatively look at features of the seabed as we pass along.
So we're essentially about 2 meters off here. We'll try
Okay.
to do 2 to 2.4 meters.
Great.
Yeah.
Are you happy with the speed?
Pardon?
Are you happy with the speed?
Yeah,
of the transect?
yeah, that's good.
Okay.
Thanks.
So we'll say this was kind of the starting area then.
Okay.
I've basically been doing it since
So we-
that green dot right there. So
Right.
put a waypoint in there if you want. For
Yeah,
endpoint.
that would be great.
Sure.
Yeah, so we're just kicking off our
I'll do
transect
it
now.
again here when my hands are free. You see where our ROV is on my screen? Yeah. You see that green dot that divides the start and waypoint 2? Just throw a waypoint right there. I can do that first. This one. Okay, yeah, the blue dot behind me will be on your screen. That's the starting point. This one? Yeah. Okay. So, just stuck up some of that dot to it.
Yeah, so the seabed we're passing over now is very dominated by sediment that's being shed from the reef above us, driven by currents and failures, mass failures along the margin of the surface reef, and sediment moving down this steep flank of the reef into many hundreds of metres of water.
me to slowly turn you up? I'll monitor it.
And it's 6 will come up. Oh no, I'm just saying just to let the kid keep up for this trend. Oh, you want us to stop?
Andy, are you ready to start speaking?
Yeah, just trying to hook this thing on again.
Yeah. So the ship's going to come down to that big waypoint? Yeah. The only thing is I can't. Oh, I'm doing that. So stay just a little bit away. So I'm going to just slow speed. Yeah, this is up cool just to give the viewer something to talk about. And we'll grab something else up.
Roger.
Yeah, you to give me an explanation.
Is it good?
Thanks.
I'll turn you up slowly here.
And they want to give your spiel, and then we're going to stop. They want us to stop along the way. We're getting too fast, so we'll collect something.
Yeah. Yeah. So I won't rush it, so it sounds silly, and then I'll close out. It'll be the blog as the closeout. So for those who want more information.
We're live.
Hello viewers young and old. My name's Andrew Carroll. I'm a marine ecologist at Geoscience Australia, and I for one feel very privileged to be participating in this journey of discovery on ROV Falkor. You might be looking at a bit of sand at the moment, but my background is in coral reef ecology and specifically coral reproduction. A little shallower than this, but it's certainly to be amazing to be back on a GBR, and even more amazing to be sitting with the team behind ROV SuBastian, and of course the many scientists involved in this research expedition, including Marty McNeil, who's sitting next to me at the moment, madly annotating and keeping log of this particular dive. So for those of you that have been watching some of SuBastian's live streams, you may have noticed that we've been doing, as part of these deeper dives, a transect like this. And it sort of appears that we fly over some of the cool stuff that we've just been looking at. We rarely sort of pause or zoom in to have a closer look at some of these things. Here's an exception. I'm not sure why we've stopped. But there we go. Part of doing these transects, there is reason behind it, and a good one at that. The Marine Biodiversity Hub, which is part of the National Environmental Research Program initiative in Australia, has developed over years a series of field manuals to ensure that the data collected by seafloor sampling platforms like ROVs or autonomous underwater vehicles at different times and places across Australia use similar methodology so that we can make some comparable observations. So for each of the ROV dives, we've dedicated a 500-metre transect, and that will be quantitatively analysed. So we have SuBastian at an elevation 2 metres above the seafloor where possible, and we travel at a consistent speed unless there's an issue where we have to pause or stop, like at the moment. And that reason is because the ship is catching up to SuBastian. We're going too quick for the ship. But what we try and do is maintain this consistent elevation and a consistent speed, and apply this method consistently across all of the reefs and deep reefs that we're visiting. So another benefit, I guess, of having these straight transects is that when we classify habitats and mark areas of transition, rocks and boulders and other hard substrata in the deep sea are premium real estate for many organisms, and pulling that information out of these transects becomes quite powerful in the sense of saying something about the habitat. here we see SuBastian having a look at a conglomerate of coral rubble, other invertebrates, while we wait for Falkor to catch up. And here's an example, I guess. You might fly over this particular patch of sand and not necessarily appreciate some of the animals that are living here. Taking these timed transect approach and having a 5-second screen capture grab allows us to build up a large image data set across this 500 metre zone, and from that we can pull quantitative information from the imagery. So that might be the presence and absence of animals of interest, like black corals, soft corals, like particular species, or it might be just general understanding of the percent cover of the benthic composition across that transect. While we're paused here, I might just pass back over to Jeremy, who's our black coral specialist. We're using the most of the time for Falkor to catch up with us to sample this particular specimen. The beauty of these transects is, with pauses such as this, we can pick up and continue where we left off. I'll just hand you back over to Jeremy. I'm sure he has something to say about this particular species.
Sorry to disrupt the transect, but as we're waiting for the Falkor to catch up to us, we spotted this awesome black coral I think in the family Stylopathidae. This looks like a Stylopathias. Perfect. So we could take a close look at the colony. So this one's really interesting. A lot of the Stylopathidae, they have what are called worm runs. And so if we could focus, you'll see worms that have modified the corallum. So the skeleton has been modified by something other than the coral itself. And this presents a challenge for taxonomy because, as you could see, the stem has this channel running through it, and so we don't know what the coral looks like in the absence of these channels, because we always find channels with this genus and these species within this genus. So we can tell what genus it is by the pinnules, the repeated branching, and the spine, the skeletal spines, that informative feature that we need to identify the species. We can't see it. So it's a challenge, but we do now have more advanced molecular techniques than we used to. So although we can't see the spines, we do have more information to try to place this species in the order, the black coral order. So it's fantastic that we found this specimen. Very opportunistic that we had to stop, otherwise we would have passed right over it. So yeah, let's put it in the box. Yeah, this is a fantastic find. Often the species in this genus are dredged up, and in addition to having a worm run, they're also tumbled by rocks when it's dredged. So rarely do we ever get this species in this genus in perfect condition. So once it comes aboard, I will photograph it, take a subsample for DNA analysis, and I'll preserve the rest of it in ethanol. And it'll be driven straight to the Museum of Tropical Queensland to be preserved. Yeah, that was really exciting. So now the ship is a little bit ahead of us, so we can levitate and head out. And so we are now continuing our transect. There's a whip black coral, it seems. There will be plenty more whip black coral to collect. Yeah, you can never have enough cameras on this ROV. How many cameras are on the ROV?
Plus Googles.
We got 9 cameras on the ROV. 14 total. So we have
stereo
the
for
ROV
Google.
cameras, we have stereo cameras on the ROV. So we do have plenty of cameras, and we are recording on all of the different cameras. So, although you can't see it, we've got a lot of images and videos being recorded.
Want me to get one of you about it?
As this transect continues, you can see a lot of stuff going on. You can see lots of corals. Whip corals and some. Yeah, lots of little corals in this area. That might be a soft coral. Here's a whip coral. Looks like... it's hard to say. Yep, fern. That's the main goal. So the main goal, we're all on the same page. What we want to do is conserve the habitats and the species living in the Coral Sea and the Great Barrier Reef. As we all know, there are lots of threats towards biodiversity, and this basic fundamental biological data will allow us to make much better decisions to preserve all the life that we're seeing, really for the first time in this part of the Coral Sea. That looks like another black coral in the bottom right. This looks like a soft coral in orange.
Roger that. And if you could slow it down just a hair, let's catch up. Yeah, slow it down.
See a little fish swimming in the center, under a hole. There it is.
Brooke, the last week one?
I think J-Rob may have put the episode as well if you want to bring up the chart.
Yeah.
Yeah. Over the next 3 weeks, we will have guest speakers. We'll have people zoom in and talk about their expertise. So Kai will be speaking. Other people who will be speaking, Merrick Ekins, sponge curator at the Queensland Museum, Tom Bridge, coral curator at the Museum of Tropical Queensland.
Dougal.
And Dougal Lindsay. And Rob Beeman will be on the second leg of this expedition, who's just knowledgeable about basically everything. He's a great person to have on these kinds of expeditions. That's okay. That's okay. That distance.
Yeah. Yeah, you just curve out a little bit. We're going to start heading back over to
Okay.
I'm assuming these lines are guidelines.
As with everything.
Yeah, you see something too that's really interesting. You want us to stop this other stuff.
Okay. So we're slowly getting shallower, and when we go from 300 meters to 200 meters and shallower, we get quite different assemblages, and that change, that turnover is pretty distinct. And that's actually one of the things that we're interested in observing is what are all the living things down at 2,000 meters and 1,000 meters. And then at 500 meters, there are certain abiotic factors that consistently change. Sunlight goes away with depth, temperature decreases with depth. So there are these distinct changes, and that's reflected in the biology that we see. So categorizing that is one of the aims of this expedition. Well, I see a fish in the background. I think we'll catch up to it. It's a green fish. Oh.
Yeah, absolutely. Do you really care? Wow, look at that. I saw that in the other dials.
That was, I think, a really big sea pen.
Whenever you're ready.
All right,
Anywhere that you can get anything from around this edge here.
we're gonna.
Break it off.
It's got to be broken off so we know that it was an in situ rock, not some random boulder that we don't know where it came from.
Okay, sure.
So we're pausing the transect, and we're gonna collect a rock.
Anywhere.
And
See? It's ready.
She wants to break it off the edge of the
yep, so this dive has lifespan about 20 minutes. So we're gonna collect this rock and then see if there's anything else interesting nearby to collect from, and then we'll finish our transect.
Marty, yeah. Can I break it first?
Yep. You can break it off and then pick it up.
The depth is 218 meters, and the temperature is 20.5 degrees Celsius.
It's hard. Might have to try to handle it. Just purchase your delicate.
Doing a great job, man.
for dinner.
Turkey. Cooking cauliflower, chicken cauliflower, pork and ice cream. Ice cream and mascarpone dates. Coconut, pork, sauce curry. Really good mushrooms if you're into that.
It's pretty hard.
Yeah.
Let's try this one.
Okay, to finish off the dive, we have Cody and Ben at the controls.
It's gonna be Chris.
What? I mean, ice cream already.
If no one wants to drive, I'll drive.
Either I can want to leave
That's great.
it. Wait, they're not down there too.
Is that mint?
Yeah.
Mint chocolate.
Oh boy.
There's double caramel stuff. Yeah, yeah. It's regular, it's almond.
Oh, yeah.
Cone, like, don't eat that.
Oh, that's good.
Got this one, Marty.
It's okay.
We need some dynamite, I think.
It's just rubble.
Yeah, it's got to
Yeah,
be.
sorry,
Yeah, it's
sorry.
rubble.
No, it's just rubble.
Yeah, you gotta go through it.
Yeah.
This piece? This big one?
Yeah, we need it on the exposed face of a big bit about there.
Maybe.
The bridge.
So maybe this explain Brittle.
Yeah, that one, maybe.
What the target is?
So it's going to take over.
Yeah.
Then you can't have Western go to dinner though. Not barbarians.
Ah,
Yeah, I got it.
Jeremy, should I explain what we're trying to do?
So right there.
Right.
That bridge.
At the moment, our ROV pilots are trying to position us in a spot where we can bust off a piece of the exposed limestone deposit here. These look like massive slope failures that have debris flows that have come off the edge of the reef. Yeah, quite a while ago. They've since been lithified, turned into rock, and as you can see, very very heavily eroded and bio-eroded as well. Lots of boring organisms have been using the rock as a habitat. Sebastian's robotic arm is just positioning itself in a spot that hopefully we can break a piece of rock off.
I'll stop pictures. Yeah.
Pretty far off. We're on the core right now.
Good to know that. 14.
So the two green dots you can see are the laser markers, so they're around 10 centimetres apart.
Did I hammer it? The wrist don't snap it. What's that? The wrist don't snap it? Yeah, no, the wrist one's in it. Looks like working at all, actually. What? Yeah.
And that's the end of our dive this afternoon. We're now heading back up to the surface and back onto research vessel Falkor. So thanks everyone for joining us, and we'll be back online most likely on either Wednesday afternoon or Thursday morning this week. Thanks for joining us.